Hello, dear guests. Welcome to our meeting. And my name is Esra Cebeci, and I am working as an eggplant breeder at BATEM Agricultural Research Institute. And in here, we have been conducted a European Union project for 3 years, titled Crops for Change, Tackling the Global Warming Effects in Crops. And this project started in 2021 with our valuable partners from Italy, Morocco, Algeria, and South Africa. Since 2021, we did research on improvement of abiotic stress tolerance of rice, wheat, and eggplant. Today, we would like to share what we did in the frame of the project for these years with you. And before start, I would like to thank all of you who join us here with our foreigner guests. And there is one more thing before start. Me and my colleagues wants to share a video presentation, which is a pretty short but full of information about our institute. Please enjoy your time. I will see you after video presentation.
Establishment of Batı Akdeniz Agricultural Research Institute, that is abbreviated as BATEM, is dated to the first years of the Republic of Turkey. The institute took its final form by merging of 7 institutes in different times. BATEM is the biggest institute in Turkey and responsible for West Mediterranean region. Scientific researches are carried out in field crops, fruits, vegetables and ornamental plants, food technology and medicinal plants, soil and water resources, plant protection, agricultural economics, training, and dissemination. The institute, which carries out its researches in 2,856 acres of open area, 41 acres of greenhouse, and 4 acres of laboratories in 5 different locations, is among the leading organizations in the food and agricultural sector with its national and international projects. Until today, almost 90 different cultivars were developed in the institute as a result of citrus and subtropical fruits breeding studies. In the tissue culture laboratory of the institute, 300 to 350,000 virus and virus-like diseases-free budfoods are produced annually with the shoot-tip grafting technique in citrus fruits. Around 60 field crop cultivars were developed as a result of cultivar development studies on warm season cereals, cool season cereals, industrial crops, and forage crops. Besides seed production of the developed cultivars and cultivar registration trials, research and development experiments about agricultural mechanization are conducted. In 2010, Medicinal and Aromatic Plants Central Laboratory was established in the Institute to create an infrastructure for the researches needed in the food and health sector in the field of medicinal and aromatic plants. With its modern and technological infrastructure, research studies are carried out in the fields of identification, conservation, cultivation, characterization, and evaluation of medicinal and aromatic plants richness of Turkey. Moreover, analysis services are provided to the research institutes, universities, private sector, and all relevant institutions and organizations. BATEM has the first registered carnation cultivar in Turkey. Also, researches are conducted on indoor and outdoor plants, as well as cut flowers and bulbous natural flower species suitable for open field and greenhouse cultivation. Studies are carried out on breeding, cultivation techniques, and the collection and conservation of genetic resources in endemic ornamental plant species found in Turkey, especially in the Western Mediterranean region. BATEM carries out research and development studies on breeding, cultivation techniques, collection and conservation of genetic resources, and greenhouse technologies in vegetable species suitable for greenhouse cultivation. Also, the Institute developed and registered nearly 40 vegetable cultivars. The cultivar development studies carried out under the coordination of the Institute contributed greatly to the increase in the use of local hybrid cultivars from 10% to the over 50% in the vegetable cultivation. In the pilot food production facility of the Institute, besides citrus peel jam, which is traditional for almost 50 years, eggplant, watermelon peel, fig, pumpkin, date fruit, tomato and kumquat jams, citrus marmalade, concentrated orange juice, bergamot powder and extract, pomegranate juice and syrup, dried pomegranate seed and pomegranate seed powder, medicinal and aromatic herb teas and oils are produced and sold. Every year, virus-free 60,000 citrus saplings, 10,000 avocado saplings, and 10,000 pecan saplings are produced. Moreover, pomegranate and loquat saplings are produced on request. Plant Health Laboratory conducts research and development studies and provides diagnosis of plant pests and diseases, also controlling methods and plant health clinical services. Every year, 1 to 1.5 million predator and 2 to 2.5 million parasitoids are produced and introduced to the citrus and pomegranate growers for using them as natural enemy of citrus mealybug, that is the most common pest of citrus. Leaf, soil, water, and organic material analysis is done in the Soil and Water Resources Laboratory. Moreover, fertilization schedule is prepared for the plants that are mostly grown in the region. This is the only laboratory in Antalya city for organic and organomineral fertilizer analysis within the scope of regulation of fertilization. The institute makes economical analysis of conducted projects and socio-economic analysis of agriculture enterprises. BATEM organizes demonstration studies, theoretical and applied training activities, fairs and field days in cooperation with other public institutions, universities, and private sector, so that all these research results can reach the producers and related companies in the sector. The Institute's journal, named Horticultural Studies, is indexed by DOAJ, FAO Agris, TÜBİTAK Ulakbilim, OpenAir, Index Copernicus, and Google Scholar. BATEM, which has contributed greatly to the country's economy with all these research and development studies, has developed approximately 160 registered cultivars. Today, 80% of citrus cultivars, 85% of pomegranate cultivars, 70% of sesame cultivars, 80% of peanut cultivars, 50% of popcorn cultivars, and all loquat, pecan, and avocado cultivars grown in the country have been brought into the sector by BATEM. BATEM continues its scientific studies with its researchers and technical staff to make agriculture a profitable business line and to increase the contribution of agriculture to the country's economy.
Hello. Thanks a lot, Ted. And so now I would like to invite our project leader, our project coordinator, Dr. Giuseppe Rossino, to lead this workshop. And the doctor is working on breeding vegetables on Council for Agricultural Research and Economics in Italy. Please, Giuseppe.
Thank you to Felix and the Turkish partner of our C4C project. This is a project funded by the FOSC, EC Eranet calls that was finalized about the climate change. So the resistance, generally the work, scientific work on abiotic stress. The origin of this project was just talking with Ignazio, that we talk a lot about the possibility to apply to this call, and we decide to do what we are able to do. So the part of cereal for him and vegetables, and in particular eggplant for CREA. And then what we need to have other partners. We had a long, let's say, very long tradition of collaboration with BATEM, with the group of BATEM working on eggplant. Then the South Africa, Moroccan, and Algeria partner joined to the project, and we set up this proposal. Unfortunately, just after starting, we had the problem about COVID. This makes some limitation to the project, to knowing each other, to carry maybe better the work that we planned to do. But in any case, today we have at last the possibility to meet each other and almost all the partner and to share the results. As some of these will be also presented from the partner. The begin and we thank also the BATEM partner to organize this workshop. There is a general part with some speech, and then one more specific, but still general, I hope, to share the activity and the idea that came out from the project with you, with your question. So I would say, just to start the first section, inviting Felix Boyage to talk about the impact of global climate change on agricultural production. Each speaker will talk, make the presentation from this chair. Please.
Ladies and gentlemen, good afternoon and welcome to this workshop organized by BATEM and CREA. First of all, I would like to thank our project coordinator, Dr. Giuseppe Rotino, for organizing final meeting in Antalya and giving opportunity to meet our international partners. I would also like thank to Directorate of BATEM and to Project Turkey representative Dr. Esra Cebeci for their wonderful hosting, and everyone who contribute to organization of this meeting. Yes, this is the question ask ourselves. What's the climate change? Climate change refers to long-term shifts in temperatures and weather patterns. Such as shifts can be natural due to activities in the sunlight or a large volcanic eruption. But since 18th, human activities have been main driver of climate change, primarily due to burning fossil fuels like coal, gas, and oil. Burning fossil fuels generate greenhouse gases emission that act like a blanket wrap around the world and trapping the sun and rising temperature. Greenhouse gas, the main gas causing climate change include carbon dioxide and methane. These come from using gasoline for driving a car or heating a building, for example. Clearing the land and cutting down the forest can also release carbon dioxide. Agricultural, oil, gas operation are major source of methane emission. Energy, industry, agriculture, land uses are among main responsible sectors of greenhouse gases. Model projection reveal a climate situation never experienced before. Global temperature is projected to warm by about 1.5 degrees Celsius in 2050 and 2 to 4 Celsius by 2100. The current global agricultural lands in the world are already insufficient to meet food demands of growing world population. Climate change is putting huge pressure on agriculture and production. If action not taken, carbon dioxide emissions are expected in remain high in coming decades, and adaptation of policies of countries most affected by food insecurity will be inadequate, and the situation will be catastrophic. The situation worsens in a devastating way in India, Southeast Asia, Africa, Central America, and also some countries in Latin America. The correlation weather-related crisis and rising hunger in these countries is stark and undeniable. Extreme weather events frequently cause severe crop yields losses and affecting food security and also farmers' income. Drought is a major environmental threat to sustainable crop production. This severity damage caused by drought is generally unpredictable as it is driven by various factors including rainfall patterns, unpredictable rainfall and moisture holding capacity of soil, and weather losses through evapotranspiration. High heat stress or high temperature in temperate regions, this shock during reproductive phase can cause substantial reduction in the yield. Heat stress causes decreased growth entire plants by affecting both root and shoot growth. Global warming progressively increases in frequency and severity of heat waves. It tends to be hotter in summer and cooler in winter. Typically, plants has a narrow climatic range correspond to optimal growing conditions. Outside this range, their growth is decreased even or collapsed. Farmers' areas accustomed to regular rainy season are now experiencing unpredictable rainfall. Sometimes this comes from usually heavy rains, which can have devastating effects, eroding topsoil, washing away nutrients, and destroying productive area. Most of the pictures belongs to Antalya, unfortunately. Change the global water cycle response in warming over 21st century will not be uniform. The contrast in precipitation between wet and dry region and between wet and dry season will increase, although there may be regional exception. Extreme precipitation events over most of the mid latitude and land masses, wet tropical regions will likely become more intense and more frequent by the end of this century, as a global mean surface of temperature increases. Nature seems to be getting more violent in recent years with increased severe and numbers of storms. Tornadoes rip the top from silos and level whole fields of green crops, destroy greenhouses, and harvested production waited to be shipped. Also, this greenhouse problems comes from Antalya again. Salinity, also the other abiotic factor, plant face to this stress. Soil salinization can be caused, including increasing evapotranspiration causing by drought season and or rising sea level in the coastal areas, and inappropriate irrigation practice using groundwater with high saline concentration salts also lead to severe soil salinization. Globally, more than 900 million hectares of lands under threat by excessive salt problem, and situation is going worsening. Soil salinization is severely detrimental environmental factors that affect seed germination, crop growth, and production. Abiotic stress possesses significant effects on plants at every stage of their life cycle, especially during growth and development. The impact of abiotic stress on plants manifests in alterations to their morphological, physiological, and biochemical process. However, this specific effect of abiotic stress vary by crop, stress type, and exposure duration. And this stress leading to diminished rates of growth, germination, photosynthesis, hormone and enzyme activities, distribution, heightened biooxidative stress, and reduction crop productivity. Finally, we must underline that climate change is a source of environmental degradation. Hence, it can even more magnify problem on agricultural production. We must bear in mind the climate change will significantly affect the duration, quality, and growing season. The latest IPCC report predicted modification in areas, sustainable food production, fresh water, as well as biodiversity. For example, impacts on pollinators, such bees, they are already under great pressure from habitat losses and intensive agriculture. The same is about effects on pests and disease in crop. Timely adaptation for future climate will shift showing dates in the extratropic. Due to the earlier spring onset, spring season show advancing earlier showing dates depending on the region. In contrast, adapted showing dates in winter crop with vernalization postponed to avoid that the crop develops too much before being dormant, which could be made more susceptible to frost damage and disease. This effect of climate change on agriculture will depend on rate and severity of change, as well as degree to which farmers and ranchers can adapt. As conclusion, to meet increasing for demand of world production, while many areas have been considered marginal as growing crops due to low fertility, salinity, drought, and many abiotic stress, now have been incorporated with production system. Climate change has brought new challenges to agriculture. Plant breeding and improvements in agronomic practice can make consistent contribution to increase global production year upon year. To cope with all these new challenge, many plant breeding programs have reoriented their breeding scope to stress tolerance in recent years. Please note your take-home message. What the global agricultural system will look like in the future will depend on what we do today. Thank you.
Thanks to Felix. Is there any question? Okay. So now I'm going to talk about the breeding strategy for plant tolerance to climate change stresses. My talk will be maybe not a classical talk, just some consideration that mainly because of this project that I coordinate during this year, doing this work that in the previous year our group didn't face on. We start to think how to manage this trait that was quite different from the previous breeding work we were doing. So we have to again thank this collaborative project, and so the scenario, the general scenario that already Felix showed is about the we need to increase the supply of staple foods and vegetables that continue this trend that started and increased a lot with the Green Revolution about two centuries ago, which was due rely generally in higher input of fertilizers. You can see here the increase in the consume of nitrogen, phosphate, and potash. The increase in the last, let's say, 20 or 25 years of the fresh water consumption for the agriculture, and agriculture is the major utilizer of water, and the the increase in the temperature that you can see also in this graphic. So our Yeah, change again. Okay. No, it's okay. So our breeding strategy is, I'm talking not about the scientific aspect, but from the way of a breeder that is looking to supply, to release a competitive variety. So what I think, or how to manage or how it should be managed in the next year I'm breeding program to face, to try at least to solve this, to help to solve this problem. So as we say, there is a need to feed an higher population, as said Felix, with the risk of malnutrition. And this is should be done both increasing the yield, so the production, the amount of production, and simultaneously at the same time reducing the use of the input, so to have a more sustainable agriculture. So and these are to take into account also the cropping system. And so the final purpose is to recognize, to identify all the beneficial alleles present in one species, to accumulate this, so to have the better variants for each allele, eliminate the deleterious variants, and try to apply the cutting edge technology to reach this goal. So I'm about all the stress that Felix talked about. I will focus mainly on drought, heat, and water shortage, but of course, this is valid also for flooding, extreme if rain, and why not for cold and other abiotic stress that were cited before. What we can say that most of these traits are complex, the so-called quantitative traits that are quite difficult to work with in a plant breeding program that has as focus to realize resilient, resistant, or tolerant varieties. So varieties mean that has to satisfy the growers, not only the scientific aspect, to have to understand better the mechanism. This is important, can be exploited, but should be used for reach to improve the possibility to have real aspect, real activity that can improve the tolerance. So because we can say that most is many years that breeding activity, especially in cereal, but now also in vegetable or other species are involved in breeding for abiotic stress resistant, drought and water shortage or heat temperature. So the fifth aspect, and I realized looking how our activity was going, is what selected for, which trait we had to consider. And so the fifth, we talk also inside our group, we have to select for absolute high yield or for the minimum yield decrement. Generally, and for this there is quite a lot of literature on cereal, we can say that the better results have been obtained selecting for absolute high yield, especially also like in wheat or other maize. So also growing this, obtaining this variety in condition where there is higher input of external supply of fertilizer or water. This genotype still of varieties still remain the higher producer when you move this variety in a condition of stress. This is quite surprising because this means that maybe we are still not understood the mechanism, the real mechanism that allow one genotype to reduce the gap from non-stressed environment when compared to unstressed environment. And even if in the higher modern cultivar there is an higher reduction of product other genotype, this total production is still higher and maybe often is even better as quality. And then the materials, the genotype in which there is a less reduction in stressed versus not stressed genotype. Generally, most of breeders say that a good material should have a gap yield, so a loss of yield of about 80% to be considered as a tolerant or resistant one. And so what to see about the breeding? The breeding is looking to chromosome, to genome, the nucleotide. So the identification, I said, as I said before, gene QTL, transcription factor, microRNA, that are all genetic factors that influence the expression of the genome. And to understand, we can generally, the researcher focus on one particular aspect of this. So you select or you inactivate or you overexpress a single gene, but the reaction to the stress is influenced by environment. And so we should plan a breeding program that should be contribute to have on-farm drought or water shortage tolerance. This means to dissect the interaction between genotype, environment, and management. Environment including both, so from my genotype point of view, we should study all the association and interaction of the network of genes that interplay and respond to the various stimuli from environment, including also the management. So to have to understand better the genome, the allele, and to to have a good genomic prediction and genomic selection of the best combination of alleles. But from the environment point of view, we can consider that we can have some region, some place where there is mainly a single factor, stressing factor. The factor stressing can be combined, can be sequential, can be multifactor, so different stress or different type that simultaneous act. And this we can say, especially like for our vegetable production that go from very different temperature from night to in protected cultivation along the season. There is very fluctuating temperature. For the and we have to consider also the biotic stress. Another thing that now is more and more considered is the soil characteristic and holobiome. So the microbiome at soil and also canopy level, that this may have a very strong influence. And also it's quite important is the management, so the cropping system employed. And this goes from plant density to agrochemicals employing, watering regime, quality of water, pruning, all the aspects of cultural practices. So we here just to see that for the genotypes we are going to reach a very deep knowledge. We have from genetic map using very, let's say, archaic markers. Now we have genomics, epigenomics, pangenome, gene discovery. We have the possibility to transfer one gene to another species or to modify very precisely the single gene at the promoter level, so influence the word to be expressed or in the coding sequence. But what we can see also here in this graphic, just before coming here, I make a search on Scopus database using as keyword climate change and plant breeding. And there were in the last 20 years more than 4,000 published peer-reviewed article that goes from only 6 in 2001 to 608 to 2023, and now we are more than almost 450. But the translation of all this very huge amount of work has been less successful, we can say, looking from the grower perspective. We have just a few examples of real improvement using transgenetics. This is an example on wheat that is a transcription factor from Elianthus annum that has been unwanted, mutated during they grow in E. coli and was transferred in wheat. And we can say that this had a very good validation at field level, because this publication here that is of 2019 was based on 37 field experiments lasted 9 years and involved 13 sites. So it's quite comprehensive. And the increase of production goes from 6% to 9.5%. And when there is stress, the benefit, so they increase the higher yield that gave just using this transgenic wheat was 60%, reaching also in 95% when there was very strong dry situation. And this is also an example of cultivated transgenic materials that has been approved for cultivation in Argentina in 2020, but last year also in Brazil and Paraguay. In the other state that you can see here, this was approved for feed or food. Another example that I think need to be, and this, the mechanism of action of this is one single gene mutated, so it's quite similar to the gene that make the low 8 of cereal, and is trigger a mechanism to avoid that the plant, when there is a water shortage, doesn't start all the mechanism of senescence. So they last waiting that the situation improve and then restart again. So, and is involved with many networks of metabolic networks, especially with abscisic acid. Another example that, in my opinion, is also similar, but maybe has to be validated further, is more recent, and is a gene-edited ROP9 tomato that in this silenced gene, they make, is involved with stomata closure, so to reduce the stomatal conductance without affecting the photosynthetic activity. And they make some large scale of one of the mutants they obtained because there was different expressivity of this trait with different lines. This is quite so, but is not so large as validation as the previous one. For all the other improvement in resistance, there is improvement, but just trying making classical breeding. We can say that there is also a reduction of this gap. I would like also to show you this paper that talk about the guideline for designing and interpreting drought experiment in controlled condition. They generally as that one the scientists will use instead of when, but I always say that the field is the final response we get in the field, not when we try in our grow chamber or we try in greenhouse. And in many paper, this we talk also in our group often. We say this line is tolerant, this is resistant, but we are studying only one single trait, like photosynthesis, relative water contact, that is just a single minimal aspect of all the reaction of the plant. So there is a problem that scientific community maybe should agree about the terminology that used. And in any case, when we study, that is necessary to do to understand what is doing each gene or how this gene interact with the other, with the metabolism. But maybe to have a prediction that there is a tolerance or resistance very often, as they say in this paper, there is misinterpretation or can give misinterpretation of the results. This is a contribution to a better understand of the functioning of some gene. So, and often it's important or the design of an experiment, so to have a good number of replication, randomized, and also it's important when and how you apply the stress, because in any case is an artificial stress that is applied. Is not the real stress that you have when you grow outside the plant. So going again with stress, we can have a mild stress that can be last longer, or very severe stress that can last lower. These are different effects on the plant and on the production. Then as I said before, the plant can combat against a single or sequential stress or multi-factor stress and overlapping stress. And what is important to have recover the plant from the mild or severe stress. And when we plan our experiment, the number of replicates and possibly monitoring the soil plant status, the microclimate variable, and now the microbiome are quite important to have information about this factor. And it's important also when and what apply and measure the stress, what kind of measure to do. So to have a morphological, physiological, biochemical, molecular survey of the reaction of the plant to the stresses. And here we are accumulated much number of data, so we have big data that need to a very high computational power to be understand, and that can help to solve the understanding, the better understanding of the reaction of the plant to the stresses. So what we can say that we need to increase the yield in any case, as we say before, to increase also the efficiency of use of fertilization and of water use efficiency. This will reduce also the input and reduce also agrochemical. So all these aspects are important, and generally
is going to make to breed a real variety faced with this problem. It's not with the single. So the very last one to produce, to have a produce that maintain quality. And this is quite important for vegetables, which are not or let's say for the produce that are not transformed like the grain, but for the vegetables or other uh, produce in which the fruit or the leaf are that one are sold in the market that the consumer buy is not are not processed. The strategy, the best one is to solve to exploit the genetic resource, so to better understand and exploit the crop germplasm, so with interspecific hybridization, using also experimental population like introgression line, Bacross advanced line. The many help can be obtained by comparative genome from one species with respect to the model species to which it belongs, exploiting orthology and synteny. The genome sequencing is going very fast and creating pangenome, so to have a better figure of all the genome organization for each species, considering the essential genome gene which make a species and the disposable gene that make the difference between the various accession. And the management of big data, as I said before, that may allow a more precise prediction. So all these omic resources may facilitate the knowledge of genetic basis and identification of candidate gene that interact and may have a role, hopefully very important, in the molecular network underpinning the traits of interest. Another aspect that scientific community are looking more ahead is de novo domestication of plant. Till now we have 3 major crop: rice, wheat, and maize. Over the 150 commercially cultivated that furnish these 3 species crops almost 60% of the calorie consumed by human beings. But there are almost 2,500 plant species that human being can or have been used or can be used. So we have a very large possibility. We can reintroduce some already semi-domesticated that are very niche cultivation in the cultivation. We can better know the variation, genetic variation in the wild species and exploit this one. We can try to increase this maybe could be also a good way to increase the perennial crops that seems could be more resilient towards some biotic and abiotic, especially abiotic crops, or even we can re-domesticate some cultivated crops, because for some of the more important cultivated species, we already know the genes that when long time ago mutated were selected by the men who start agriculture. So we could make a better re-domestication of the cultivated species. So in conclusion, I would say that for each crop, the generation line that displays simultaneous variants in several genes, using also genome editing, putatively involved in stress-related climate change effect, in this case mainly drought heat, will help to better understand and exploit the network and crosstalk mechanism that underlie tolerance to this trait. And because we have the possibility, we talk also this morning about this aspect, when we consider two different accessions, the genetic difference between these two accessions is very large. There is a very huge difference in the genomic composition of the accession. But when we can generate the mutated in several genes with the same background, genetic background, we can attribute this difference to the network or networks that we try to modify. So to have a more a better knowledge of what we of the result we obtained. And with this, I've finished. And again, thanks the project that give us the possibility to think about this aspect. Thank you. If there are questions. Yeah.
Well, first of all, thank you very much for this wonderful talk. And at the end of your presentation, you talk about the importance of using a big data when it comes to better prediction, like more precise prediction. So, and also you talk about the advantage of transgenic plants, especially its benefit in getting more yield under the drought conditions. And mainly you talk about the genomic prediction as well. So what are the additional data we can use for better prediction in either wheat or eggplant? So I'm just wondering what are the additional data instead of additional data, different data than genome data? So what do you suggest? And also, what do you mean by saying big data for better precision? So I'm able to about that. So thank you.
This is what I understand that are quite important, because now most of breeding you do on the computers, and I came from classical breeding, so from the field, but and it's I think a better interaction between who collect data also from environment. There are can be let's say correlated or used together with the molecular data to try to design a model, to a prediction, and we have also the possibility to verify with using genetic editing, maybe taking information from different independent correlation, let's say, but I don't know if is right the terminology, and verify if it is work or we make a step ahead in the understanding of this. I don't know if was this is my idea about this.
Benim İngilizcem bu soruyu İngilizce sormak için yeterli değil. O yüzden arkadaşlardan translate etmesini rica
I'll try to help with the translation. He was very impressed with your presentation about the three main cereal crops. Yes. Okay.
kontrollü su, kontrollü gübre, kontrollü ortamda ne gibi stratejilere, özellikle yeni çeşit geliştirirken bu solanus kutucuklerde ne gibi stratejilere dikkat etmemiz gerekiyor?
Okay, that was very long. Okay, I'll try to I will try to translate it as much as I remember. So we are all breeding vegetable crops, and especially for greenhouse growers. He was wondering what are the strategies that we can employ in means of irrigation, fertilization, and other things that we can do. What should be our main strategy for breeding vegetable crops, specifically for greenhouse growing?
Yeah. Also for greenhouse, I think the same measure idea to lower the input, but so maybe to think different architecture of the plant also, the possibility to better exploit, to improve, I don't know, photosynthesis, to improve and monitor also the supply of fertilizer. For instance, we had another project in Sicily about this aspect, and when we make analysis of water and soil, we say there is too much, both in soil and water. And we oblige the grower for scientific reason to not just to give, not to add anything to the water that he pick up from the well, and they grow normally. He was quite surprised. So maybe all the society should look to to this aspect, not to necessity to sell the products, to grow the plants. And but as I said before, for vegetables and Solanacea or Cucurbitacea, what is then important is the appearance and quality of the product, that this is on top of everything. So we have to change, to reduce, but maintaining the level, the quality level of the production that we have, or even improve it with some nutraceutical or benefit for the human health. I don't know if it's then okay. I think some changing in the way of growing the plant. We have soilless culture. We have a way of pruning or that is more or less adapt to one genotype to another. Maybe we have to, when we make this activity, to think also to the reduction of input, still maintaining the quality and the yield, or just not reduce it.
Thank you.
Another question?
Hello,
Hi.
thank you for presentation. I am Mustafa. I am molecular biologist and genetics. I understood the molecular mechanism, also genetics of cell plant, but there are a big and different issue in this area. I guess this is the epigenetics. Epigenetics, I guess, the most important area that we can focus on, because we can solve many different problems in genetics, but epigenetics, I guess, huge and dark area to understand how different environmental factor affects cell and plant, especially. In our company, we do mass selection, but we must improve this area because manual breeding is left behind, I guess. As breeders here especially must understand molecular biology and transfer the technology, invest on genetics, because Mandel breeding is time consuming and trait does not pass directly to the other yes the other
the other generation.
Yeah, the other generation. What do you think about the epigenetics?
Actually, there should be, or I don't know if then I can sell this. Among the omics, also epigenomics has its importance. There is this stress memory in this case that can go to, from one generation to another, even if the structural genome is not affected. And this is quite important. And maybe this is, not maybe, this is another scenario that we should try to better understand and exploit. I agree that also this aspect, I think that I wrote also something about epigenomics that became a more important field of research. But again, we should make big efforts to translate this knowledge in actual results. I think you no, you know.
Okay. Yeah, I'm in the group, but I should, because I ask him all the time questions, but I will ask him this question. So yeah, when you say the stress index, because it depends with who you are talking. If you are talking with the grower, for him, I think the stress index is the yield and the quality, and it's the cumulative effect. Sometimes I think, like for physiologists, we do some mistake because when we measure the fifth leaf or the 10th leaf, but we don't have an idea about what happens for the crop. Sometimes if you stress your crop, probably is good for the yield. So here we are not talking about stress. So I think I agree that the scientific community should probably take in consideration what are the key of stress for the crop. This is one point. And the second point, like as a breeder or as people working in genetic, because I'm agronomist, so I think it's impossible to fix like the stress factor. In the growth chamber, yes, you can control, but the growth chamber is not the farming.
Even in
The
the
farm.
coffee shop.
Yeah, but
Sometimes
you can
don't
control
have-
it better in
Yeah.
the greenhouses, but in the field you cannot control it. Just for example, the wind speed. The wind speed, wind orientation. It can change your stomata closure and opening at 45, probably better than degrees Celsius, you know. Anyway, it's a very hard
Yeah.
answer to this question. Thank you.
Yes, I agree with you that some kind of stress are exploited by, or the reaction of the plant to some kind of stress are exploited by growers to get or improve the production, to have an higher or a better quality of production. And for instance, and this is also as an example from tradition, cultivation of fico d'India or Puncia fico d'India, that they do away the first flowers and keep the other one because make bigger. Another example is lemon. Lemon we induce, or now they don't do this, but there is a particular cultivar that was very responsive to this, that in summertime they stop irrigation. The plant were, if you look the plant, the leaf were very almost dead. They became round like a cigar, and then give water. The reaction was a very huge flowering, and make these flowers, make this fruit production that was called, is called by Sardone, that is very appreciated because they harvest in the next year in the summertime when the price is higher and the quality was actually better of this one. So there are several examples, and maybe we can look to also to this or study to understand, to exploit this aspect. With regard to the grow chamber, greenhouse, open field, as I said, grow chamber, greenhouse are for according, depending on the species, are both or just grow chamber artificial condition in any case, because in grow chamber you have pots, and the pots is different from a soil, and that influence. And even, for instance, Laura can say that in our grow chamber, the wind movement is not so regular. There are some places in which, in fact, one aspect for instance, I make another experiment in which, but this was in greenhouse, in which they make mobile replication, just turning the pot to try to have more or less the same or expose. But this was done in time in different times along the season. So there is another variables, but we are facing with all this, but the final results is to have repeated and repeated trials and verify in very different situation if the reaction and the stability of the reaction is happen, the good reaction, regardless most of the stress you apply. So our goal is this. So I think now it's better to go ahead and move to the session 3, that is using genetic mapping and genomics approach for understanding and improving drought tolerance in eggplant. Is Laura Toppino is a researcher that is working with me in our activity.
Is it your fault? They told me that now I have to run. No, we run out of time. So good afternoon to everyone. I'm Laura Toppino. I'm a researcher at the CREAS Research Center of Genomic and Bioinformatics in Montanazzo Lombardo. And now I will talk you a little bit more about the genetic point of view in trying to understanding the drought tolerance in eggplant. Talking you about what we did within the C4C project. And it's not moving. Questo? No? This one? Thanks. So with the identification of intensification of the climate change, we already told that the world area suffering from drought and heat temperature is forecasted to dramatically increase, as well as the number of crops which will suffer for this stress, at least in one less part of their life cycle. And drought and heat stress both affect the reproductive processes of the plant, hampering the normal fruit or seed production and development, reducing crop yields, and affecting the fruit or grain quality. And also in eggplant, what happened? This is a short overview of the major breeding games for eggplant improvement. I've divided it into many. There are many, and I divided in breeding games from the consumer point of view, which want a fruit quality, and from the producer point of view, which want a better cultivation, better production, improved yield, but also, and most of all, it wants an improved resistance or tolerance to biotic and abiotic stresses, including drought or heat or salt stresses. So I want to talk about the indirect strategies to improve the yield or the performance of eggplant from an agronomical point of view, because we already talked about the great improvement we can do, maybe working on the water management or soil management, but among the direct strategies of breeding for the development of more resistant or tolerant varieties,
the identification, characterization, or employment of resilient accessions, and or mainly on the use of tolerant wild species. That means exploring and exploiting the variability of eggplant or another species, both between species, considering the wild and allied relatives, and within the species. So looking at the variability of the Solanum melongena species. And I talked just a little bit about the interspecific variability because later Ezra will beautifully talk about it. But the close relative species of Melongena are known to can resistance traits to biotic and abiotic stresses, and they could be employed for the integration of useful traits, including, for example, the resistance to salt or to biotic stresses of fungi, and also to drought stress, because Solanum incanum is known to carry a high tolerance to drought stress, and they can be introgressed in eggplant progenies through, in this case, conventional breeding techniques. But the use of wild species has only one major constraint, because you don't have to forget that the use of wild species may be risky because you carry on by introgressing the traits useful for your breeding, also some undesired traits, including, for example, when we identify the progeny of Solanum lineanum, for example, he carried on by linkage a high level of glycoalkaloid content. So this is a major constraint of the use of wild species when you do introgression breeding, for example. So in this project, we mainly focused on exploring and exploiting the intraspecific variability of the species. So we remain within the Solanum melongena accessions. It is known to have a large phenotypic variability, but light is low and low molecular intraspecific variability. And the neurogenetic base of eggplant is in many times a severe constraint for the development of highly performing cultivars able to contribute to increasing yield, also under the effect on environmental changes, including the drought stress. But generally, eggplant is considered a not quite, but medium resilient species to water debridement. Although it is reported mainly that a water shortage that more than 50% is causative of severe yield. But under this level, it is that are not so convincing, but more than 50% of water shortage are causative of severe yield losses. So what you can do when you want to explore the variability within a species, you can do two different approaches. One of them is called association mapping approach. So you explore the maximum amount of accessions you have available, and then you try to identify non-random association of alleles with respect to the tolerance to some stress, for example. And it is an experiment. We have a high level of diversity, so you have high marker polymorphism, you have so many traits to consider, so it is very powerful in giving you quantitative traits associated to a trait, for example. It is a less controlled experiment because you have to consider different accessions, so there is a lot of genetic variability. So at CREA, we own a eggplant collection of more than 200 eggplant accessions from two major centers of diversity. Great part is from Southeast Asia, India, China, Indonesia, Indochina, and great part of the Mediterranean basin. And we already developed and characterized and characterized also at molecular level a core collection of 191 accessions. So what we did, we wanted to look at the drought stress response, and it is a beautiful protocol for the assessment of the response to severe stress performed during a similar project, because it is a European G2P Sol project in which we were involved with. And we optimized the protocol for a severe stress assessment in our population, also because we have 50 accessions in common between this experiment and our experiment, so we can compare results. And what we did within the C4C project was to we explored our available collection. Now we explored more than 150 accessions. One experiment is still running these days in our phytotron conditions. And we assessed all the accessions for the response to severe stress of water deprivation. So after a full watering, we simply didn't water them anymore, looking at what happens during during the following days. So we assessed phenotypic data of stress index, but also relative water content index, and looking for the progress of this symptomatology. And now we collected 100 of data, which are being used in these days. Sorry for not having results still, but they are still mapping. And for an GWAS analysis to identify loci associated with this particular severe stress. I know we are in phytotrons, the plantlets are at the 5th leaves level, so it's not field condition, it's more than nursery condition. But if you want to explore a huge variability for a stress, you can think to put all 150 accessions in a field and assess them for a water deprivement, because inaffordable from a practical point of view. So we started from this kind of analysis. I know the mechanism couldn't be the same, but maybe we can select some material that it is extremely interesting also for breeding. So results are in line with the experiment which was conducted in the other European experiment. So our 40 accessions behave the same, almost the same within the two experiments. So we can rely on all the genotypic data and the phenotypic data already put in that experiment. And just to have an extremely standardized system, we worked in phytotron with always the same condition, because we tested 55 accessions at each trial, maintaining 2 accessions as reference every trial. So we have a internal standardization of results. And these are preliminary results. This is the trend of symptoms of the best 10 accessions we identified, which are highlighted in green, and the worst accession identified in red. But from a general point of view, just to summarize it, the extremely great number of results we obtained is that in this kind of assessment, most part of the accessions start to display symptoms after 4 or 5 days after water deprivation. Also, if there are accessions that after 2 days already are suffering from that stress. But the most important thing is that we identified some accessions that not working. Here it is. Sometimes it simply doesn't go. So this accession was assessed at 11 days after the water deprivement, and it is still almost without symptoms. So there is, we identified a variation between these accessions, and we identified some very interesting accessions that could be of interest for starting a new breeding programs for an improved resilient to at least water deprivement. And they can be exploited in breeding programs. But all this data for the starting of symptoms, the end of symptoms, the day in which the maximum difference variability has been assessed, which is around the 9th, 10th days of evaluation, can be utilized for QTL mapping. And that's why we are looking at it. And the most diverging lines can be utilized and selected to produce ad hoc population, segregating population for a very specific new study for the identification of QTLs associated to the tolerance. And it is the second way you can perform a QTL mapping approach, which is a biparental QTL mapping approach. So you take, instead of the entire variability available, you take only 2 male and female accessions, which are very diverging for a trait, and then you can, for example, have a more specific mapping approach. The experimental is more controlled because you have only 2 accessions, so you can identify only few traits, but you have more powerful results, and it is our next goal. So, for example, you can see this is only for example, if you have 2 extremely divergent accessions, it is easy also to identify the traits. So it is more reliable, but you need 2 very divergent accessions to do this. So now we selected divergent accessions, and we are performing something like this kind of mapping. And the final stage of this project will be, for example, the utilization of new strategies for SEC mapping, for example, which is called BSA SEC mapping, in which when you have 2 diverging lines or you have sequencing data of these 2 diverging lines, you can identify specific traits in a segregant population associated with a trait. So it is much more easy to identify, it is more easy to detect the traits associated with a trait. And this is an example of what we performed for another trait, which is, for example, associated with a resistance to a fungus. And we identified the genetic region associated with the resistance because sensitive and tolerant accession, F2 population accessions divided because they are tolerant or sensitive, doesn't map or map against the reference genome. So it is easy to identify genetic region associated to a specific traits. But as we didn't want to stuck to the lab, because we already assessed that one of the major constraints in when you go from the lab to the field, the core collection of the most divergent accessions were tested, were shared with our partner, which will talk about this in his beautiful presentation, and were assessed also at field level, just to see what happens when you go from a lab point of view to a open field condition. And the same accession, just because we weren't only in one field, were assessed also at BATEM field. So we have 2 different fields, the same core collection. So we can see, we are trying to see if there is something that can connect our result with the result obtained both in Turkey and in Morocco. So I'm too gentle with your laptop. It's not moving. Okay. Have to be rougher. So just to go with other strategies, well, I'm more interested in the development of all the genetic tools useful to assist breeding. So I'm a vegetable engineer. So I like to study markers for mass selection, mapping of resistant loci, but also more genetic studies, as this for example, RNA-seq experiment or gene editing. So this is our second part of the project, and we set up an experiment for RNA-seq analysis, taking the most diverging accession we identified, which are a reference line for which we have a genome, the other parental of a genetic map, a mutant of this line, which is dwarf, but more sensitive, more tolerant to drought stress, and other 2 divergent lines. And we made a detailed characterization of the adaptability of drought, of water, low watering condition, not drought stress, low watering condition. And we set up an experiment of half watering condition, both in phytotron and greenhouses, looking at all the phenotypic point of view, all at the assessment of all the stress indexes, not the production, because the plants are not for real production, but we also observed the flower setting, the earliness of flower setting, and the fruit set. And this is also in the greenhouse. In greenhouse was better because the light was different and the experiment went better. We assessed this experiment 3 times just to assess the control condition. Then we performed the half watering. So this is one of the major constraints, to set up the perfect condition also in lab condition. Think about field condition. So, and RNA-seq, at the moment we are extracting RNA, the RNA-seq experiment is ongoing. So we will look at the genetic network. This is what I have already explained. So the last task in our C4C project was a more advanced genetic task, because we have to study the effect of editing through CRISPR-Cas gene editing of 6 candidate genes selected from literature, because in eggplant there is nothing, and look at the effect of this editing. The genes were selected because they were at least effective in tomato, 2 of them in Arabidopsis thaliana, but now the effect is also assessed in rice. Some of them are very well known, because the major one is MIB60, which is has been already assessed to be involved in the stomatal closure. So it's clearly involved in the drought stress resilience. Host2 still involved in stomatal closure. L1 is involved in the biosynthesis of pipeolic acid. I have reference for all these genes. Another one, SDG33, has been assessed as involved in drought resilience in tomato. And adding to this one, there is also ROP9, because it was not included in this project, but we're also editing this line, looking at the effect. And at the moment, we are almost ready for the characterization of the T1 edited progenies, because we have to produce T1 progenies Cas9 negative. So at the moment we have the seed of this progeny, and we are starting the assessment of their effect. And for MIB60, the first effect of the first mutant in greenhouse in this moment, today, is that the edited T1 plants sown at the same moment with respect to their background are 100% bigger, stronger, and taller. The photo maybe is not so no, there's no pointer, but they are already much bigger. So we are confident they will have also a better response to a stress. And for the other ones, we are still at T1 obtainment, but looking for the progress. And with this, I thank you and my presentation.
Are there any questions? One question.
You're welcome. Yeah, yes, no problem.
There is the microphone.
Thank you. My name is Ahmet. I'm a vegetable breeder here, working on peppers. Also, we are likely to start a project similar to yours, drought tolerance project in our population. So I remember that you applied less water to mimic drought conditions, right, to your core collection?
Yes.
Have you heard of a PEG, a chemical?
Yeah, there are some also some publications, some papers about the PEG use to mimic drought stress condition. Yes, I.
Have you tried it? So I'm
No.
new in these areas. That's why I'm asking. I'm trying to select a methodology.
Depends on what you're looking for, because back condition has to stay. I think they have to remain in. You can test them when they are in soil if you add back. So you have to remain in more lab condition. When they are in soil and you perform a water debridement, maybe is more like the common condition. So I think that PEG would be extremely controlled condition. So you have to assess from a genetic point of view the better response. Maybe you can start with PEG, but it is not a true field condition. And also this is not a true field condition, but it is even more not true field condition. So it depends on what you're looking for.
I see. Okay, thank you.
So many papers start with
Yes,
this.
I'm confused. Yeah, that's why I'm asking.
No. It's a nice point of view, and it is maybe the preliminary can be done with this. Also in this project, in the C2P
Yeah, I was even thinking about comparing two different methods, like PEG, use of PEG, and like just use soil and less water. What are you using as the grow media, by the way? I missed the
grow media in the phytotron was soil. Was
regular soil?
No,
Not like compost or?
Yeah, peat soil.
Peat and soil, half and half.
Okay.
Because only peat is too much for soil is too you can't avoid flooding, so
Yeah.
half and half.
Okay. Thank you.
Hello. I
Question.
Sorry, didn't see you.
You see?
Yeah.
Yeah. We want some characteristic for example, eggplant. We want a high nutrition uptake, and we do this by omics technology or CRISPR. We get a desired trait by CRISPR, for example, but we get desired trait, but this trait may change the other characteristic of plant, I guess, because we ensure, for example, resistance to some virus, but we change the other part of the genes.
Because from a not scientific, a scientific point of view, we are always thinking that if you change one gene, you only change one gene. From a more philosophic point of view, the
Yes.
gene network is a net. So when you change one gene, you have to look at the effect of this changement has on the entire balance of adjacents, because also you are aware from a publication that a gene is involved in one process, maybe because it has this kind of action, but the same action may have an effect also on something that you are not expecting to. So that's why you have always to check the material you obtained, what you obtained, was the effect of the material you obtained, if you didn't get it worse when you changed it. But if you want to make an improvement, it is a risk. But also if you cross two different adjacents, it is a risky, because maybe you want a trait, but you get with drag also something else. And even worse, if you start from a wild species, you can take some undesired trait with linkage drag. So it is always a risk balance.
Complicated.
It's complicated. It's more complicated. But if you edit only one gene, I'm an engineer. So if you edit only one gene, you at least you know that the effect you gain is caused only by this gene. At least if you don't have side effect, but only one gene. So this gene causes this effect. It's only on. If you have off target, you have to check for off target. But if you don't have off target, is this gene involved in this process as its effect? It's the most controlled condition you may have.
Thank you.
You're welcome.
So let's move to the next presentation that is still about eggplant, is Ezra talking about improving drought and high temperature tolerance in eggplant by gene transfer from wild relatives.
Dear participants, hello again. And as Dr. Rotino said, I'm talking about improving of drought and high temperature tolerance in eggplant by gene transfer from white relatives. Yes, my presentation plan, you can see in slides. I'm moving. And today, one of the most important concerns in agriculture is increasing the effects of abiotic stress factors. And among the major crops, vegetables are now more sensitive to abiotic stresses. And common eggplant is widely grown in temperate and tropical Asian countries around the Middle East and the Mediterranean basin. It's a major food in human nutrition around the temperate and tropical Asian and African countries. And many of these areas have already been suffered from the climate change effects. And the project C4C, Crops for Change, which is supported by European Union post-Eranet co-fund on food systems and agriculture and climate program. And under this project, we are studying on processing to improve heat and drought tolerance of eggplant by gene transfer from white relatives. And as most of you know, eggplant needs temperature ranged from 22 and 30 Celsius degrees, and regular irrigation is needed to economic yield. High temperature above 38 degrees Celsius can seriously inhibit seedling growth, flower development, and declines the yield of eggplant. Every increase of 1 Celsius degrees during the various phases of plant growth and development cause 10 to 15% of damages on crops. Heat and drought separately or combined are among the strongest widespread abiotic stresses, and which can raise agricultural losses and reduce the crop productivity and yield, leading to lower income for farmers. Although eggplant represents moderate tolerance to abiotic stresses, heat and drought, either individually or combined, cause significant yield and quality losses. And tolerance was found in several accessions of crop white relatives of eggplants, such as Solanum anguvi, Solanum dasyphilum, Solanum incanum, and Sissimyrifolium. And interspecific hybrids of the three former species also show tolerance under different environments. Interspecific hybridization between Solanum melongena and the Solanum insanum was in our study. Interspecific hybridization between Solanum melongena and Solanum insanum was achieved first, and later drought and heat tests from F2 level were performed to the F6 level in pots and in greenhouse respectively, and tolerant lines were determined and salved to generate next population every season. And heat and or drought tolerant lines will be tested for their general and special combining ability in further studies. And our method is summarized in the slide, and we are here now. We are at the F6 level, and if you can find another budget, we can move to our hybridization studies. And in our drought tests, we used a lot of parameters, and some of them is very important, and I listed them on the slides. And we are observed these parameters on normal plants and stressed plants for the statistical analysis. And besides this, we conducted psychological analysis such as malondialdehyde accumulation and furan accumulation were noted. After selection of drought tolerance, they transferred to the greenhouse conditions during summer season. In Antalya, heat-tolerant individuals were determined via observing food setting capacity of each plant. And besides this, pollens were counted, pollen viability or germination were observed. Morphological descriptors were employed to determine the features of the lines, and tolerant individuals were selected to generate next level. Some of the descriptors derived and modified from UPO by Boyaji et al. Were used to characterize of the lines in our study. And I want to share our greenhouse activities with you in the slide. And our results, when it comes to our results, major strategies combating the heat and drought effects include screening and selection of the existing germplasm and breeding new varieties having resistant abiotic stresses. And in the present study, a total of 1,600 seedlings in each level at 2 or 3 true leaf stage in pots were evaluated considering their drought tolerance capacity, and control plants irrigated at full capacity while drought treatment applied by 70% of water deficit. And in a study conducted in eggplant, water deficit applied by 70% and 50% of full capacity. However, 50% water deficit showed no significant differences in terms of glandular parameters. Development of tolerant cultures is considered as a permanent and eco-friendly solution to overcome these problems. And Solanum insanum is reported as tolerant to some abiotic stresses by various researchers. And the results of plant growth measurements and biochemical analysis indicate that Solanum insanum has more tolerance to drought and heat stresses than Solanum melongena. And performance of segregating population derived from these hybridization generally found between the male and female parents under drought and heat stresses. We used to, as I mentioned before, we use 0.5 scale in our drought test, and we used 0.5 scale developed by Kran et al. And they are determined the genotypes with a scale value 1 as a tolerance. So considering previous studies on eggplant, seedlings show slow growth under drought stress imposed were selected as tolerant in this research. And employed parameters such as 0.5 visual scale, shoot length, stem diameter, leaf area, and so on are found sufficient to determination of drought tolerance. And generally, MDA and proline results supports the visual observations. And when media increased, proline accumulation were decreased under drought stress in the study. And while increments in proline means that has some tolerances, and increments in media means MDA means sensitivity of the plant to the stressed conditions. And in our heat temperature stress study, mean temperatures recorded above 35 degrees Celsius during the heat tolerance experiment in Sering province. And heat waves are predicted to increase, and it is of vital importance to generate crops that are tolerant to heat stress. Every path of reproductive development is affected by environmental challenges, including pollen development. And viable pollens were counted in the study. So besides this, pollen germination was observed. And during the greenhouse period, plants were irrigated regularly, and their fruit state capacity was observed. And plants were classified as heat sensitive, relatively heat tolerant, and heat tolerant plants. When it comes to take-home messages, the breeding efforts to improve the characteristics of cultivated varieties have resulted in the elimination of many genes or alleles found in wild forms, and their genes that provide tolerance to stress conditions have been unintentionally eliminated. Today, reducing the effects of environmental stress on cultivated plants by transferring alien genes from wild relatives into cultivated form is no longer as a phenomenon. And lines in the F2 generation were carried to the F6 stage through a successful testing process. And these tolerant individuals constitute a good gene pool for the development of hybrid varieties, and also will be a good source for mapping genes related to drought and heat tolerance. And it is the end of this presentation, and I would like to huge thanks for the project team. They supported in every level. They supported me in every level. I would like to many thanks to all team, and they all here now. Thank you for your efforts. And our scholars, you can see in the slides. Many thanks to come here and listen me. Thank you.
Other question? So now we will have 5 minutes for a fast coffee.
So now it's time for the presentation by John Moore about the cell wall profiling of cultivated eggplant Solanum melonga using COMP and ELISA test. John, this is online presentation, so I give the word to John.
Thanks, Giuseppe, and greetings from South Africa. So this is just going to be a summary of what my master's student Eugene did his work on. His thesis was optimization and application of an ELISA and COMP workflow for characterization of eggplant cell wall polysaccharides. So can everybody hear me? I assume so.
Yes. Well, you can even speak a little bit lower.
Oh, okay, great. My screen has frozen. Please stop the share and share again. Okay, maybe I should go through like here. Is this all right? I'm not using the full screen option. Each plant cell, and this they're composed of about 9 major monosaccharides in the plant cell wall field. And then you have glycoproteins, which are the extensins and the AGPs, which are present in the cell walls. So if you look at a cell wall, it's primarily plant cell wall. The primary plant cell wall is mainly composed of cellulose, hemicellulose, pectin, and glycoproteins, as well as the middle lamella. In the case of fleshy fruits such as eggplants and tomato, you have a very pectin-rich structure. Sorry, can you hear me? You have a very pectin-rich structure, and this is composed of different polymers, chains of galactronic acid and rhamnogalacturin 1 and 2, and you have secondary cell walls. How do we study plant cell walls? There's different approaches. The approach in my talk is going to be mainly focused on the immunological approaches using ELISA and COMP. What we do is we fractionate plant cell walls using chemical extractants, CDTA, alkali, and cadoxin to extract the different fractions, and then we probe them with antibodies specific to the specific plant cell wall components. So in this case, we have a variety of commercial antibodies available that have been generated in other labs that are available. These target different polymers that the plant cell wall is composed of, including some glycoproteins, such as the extensins and the rhamnogalactin proteins. So one of the approaches we did was to develop an in-lab ELISA for doing the assays ourselves, using enzyme-linked immunosorbent assays using an indirect ELISA to profile the using different probes to profile the cell wall extracts. And so how the ELISA protocol works is you have a target and you have primary and secondary antibodies and a developing agent. And the more target you have in your extract, the more color you'll get in your reaction. So you'll get a reaction development plate like this, the ELISA. There's also COMP, which is comprehensive microarray polymer profiling, and that's done in my collaborators in Denmark, where they use a microarray robot to print the extracts from the cell wall and then probe them with antibodies. So this antibody technology is very useful. We get a lot of information and we can get a picture of the cell wall composition. The difference between ELISA and COMP is that one is manual, one is semi-automated, one is much slower, one is more high throughput, and the equipment and training costs are different. So the aims of Eugene's study was, one, to optimize the ELISA workflow in terms of throughput and cost for studying plant cell wall polysaccharides and glycoproteins, and apply this to characterization of the eggplant cell wall. So I'm going to skip aim 1 and go through, which was to optimize reagents and protocol and automate the data processing. And so what we were able to do is generate a software program for automating the analysis of the data that we get from the ELISA and the COMP arrays. And we can generate heat maps, we can generate correlation matrices, we can generate bar charts of the different data sets, of the same data sets in different formats to interrogate the data. And the tool that we generated is available freely at GitHub for analyzing. And we're going to publish this tool soon. Aim 2 was the application of the ELISA workflow to the characterization of eggplant cell walls. So we looked at what was done on eggplants, and there's only 2 real papers in the on the topic on focusing on the cell wall components, and this was done by Paul Knox's group in Leeds about 8 years ago. And so we used material that we grew, material that we purchased, and material that we got from Korea. We looked at 3 different varieties, genetic lines, Black Beauty and 305E40 and 67.3. And we looked at leaves grown in the glasshouse, fruit, as well as leaf, stem, and root material. And we subjected that to cell wall analysis. We looked at 6-leaf old plants or 40 days after flowering, and collected leaf material and profiled the leaf material using ELISA. We also looked at the flesh and the skin of fruit, Black Beauty fruit, at harvest ripe. And then we get a profile, a heat map like this, which shows the different fractions and the different antibodies that are recognized, the material present in those fractions. So we also did COMP, and we get heat map data sets to look at. This is using local normalization. And depending on the normalization strategy you employ, you can use global or local normalization to present your data, which has advantages and disadvantages regarding interpretation. We also looked at relative levels of HEPs as a function of leaf development. We found some patterns for certain signals, which were interesting. HEP stand for arabinogalactan proteins and galactans, and these really show developmental protocol processes similarly to what it's found before in tomato and tobacco. So we got this data, and we're looking at the data. We also did monosaccharide composition, determined with gas chromatography and mass spectrometry to look at the sugar composition of the fractions. And so we are still analyzing these data sets, but planning to publish the baseline data sets soon. So this is Eugene, my student that's been working on this for the last 2 years, and we're busy writing it up. And this is his eggplant guy. And so the idea, in terms of summary of what we've achieved, so we've optimized the method for profiling food crop cell walls i.e. eggplant, and we've automated the data processing using Visualizer. We generated the biggest data set to date on eggplant cell walls. We've confirmed 31 cell wall epitopes. Previously, only 11 have been characterized in fruit, and we've looked at 3 eggplant varieties and 5 tissue types. And we've done a cost analysis. So there are limitations and future directions, the limited plant material based on for further experiments, that we need to look at all the other data sets. We need to look at the samples sent from Korea to characterize it, and from Kamal, and we're looking at other techniques to enhance what we can infer from the epitope data. So in summary, we've presented this work at conferences in Spain, and we are currently writing up the work for publication on the protocol development and the characterization of eggplant, the baseline study. And with that, I'd like to thank you for your attention, and I'm happy to answer any questions. Thank you very much.
No question. So now we move to the next presentation that is about cereal. Presented by Ignacio Edwell from University of Milan. The presentation is about molecular studies on drought tolerance in cereal.
Thank you very much for the introduction. First of all, thank you first BATEM for organizing this workshop and for hosting us for the Cross-for Change final meeting. Thank you very much also for CREA for co-organizing this workshop. And thank you very much all the researchers present today for the talk. Okay, so it's working. I cannot change the slide. Okay, so yeah, the title of my talk is Molecular Studies on Drought Tolerance in Cereals. It's quite general title, but of course we will focus on the results and outputs we have generated in the part of cereals, in the part of rice, in concrete on the project. And I invite you to follow the activities of my group in the link below on Twitter and on the research activities ResearchGate. So first of all, just I'm here for first time, I want to introduce the location of my university because we are 2 Italian partners in the project. We are located in the north part of Italy, in Lombardy, in the capital city of Lombardy, so Milan, where we have this very ancient central headquarters of the university. And we are located in another part of these campuses because the campus of Milano University is distributed along the city. So while the main campus is close to the heart of the city center, one of the most historically and artistically important building in Milano, our department is located in the east, in this part of the city, Center East, okay, where scientific faculties, research centers are located in the area of CITA study. Okay, so there are different faculties: agrarian faculty, bioscience departments with facilities for research in plant science. And in future, we will move to a new part in the north of the city, where new campuses are under construction. Okay, so I take the opportunity to invite you also to collaborate with us, to send people for exchange. Saying that, I introduce also my team. This is the seed development team. We work in development of seed with different model systems, and we use molecular function tools and also developmental biology tools to understand the functioning of seeds, the molecular pathways, the hormonal pathways that control the formation of the seed from the point of view of quality. So we use different model systems like rice as cereal, or monocotyledonian. We use also Arabidopsis, and we are starting now projects in tomato and in wheat, for instance. So the main role that UNIMI was doing in C4C was the functional characterization of key genes controlling abiotic stress adaptation in rice. So for this, different activities have been done. So at the first glance, identification of candidate genes involves in stress response. Afterwards, that identification, we start the generation of single knockout mutants with CRISPR-Cas9 technology in rice. Then we are performing actually spatiotemporal expression analysis by real-time PCR, by RNA-seq analysis on specific stress responses. And we are also doing developmental characterization of the mutants and the analysis of the responsivity of these mutants to stress. So I will quick some introductory slides because we are very short of time, but just mention that rice is very important crop in the Mediterranean basin, although we are not the main producers in the world, but rice is intrinsically linked to our culture, our agriculture, and our economy, as we have seen also in Turkey these days. And in Italy, in concrete, the north regions, the southest part of the Alps, so the Po Valley, the Piedmont, Lombardy, and Venice, are typical regions of production of rice where dishes are very important, like risotto. So during the last year, some research has been done concerning grain quality, concerning abiotic stress tolerance, and at the beginning of the project, we highlight some findings that were done by researchers working in Japan, where they discovered, for instance, that heat stress, high temperature influence the quality of grain by inducing, for instance, alterations in milling quality, generating chalky grains. Chalky grains or broken grains are affecting the quality of grain, and it is induced always during the formation of the grain when the plant suffers from heat or drought. Okay, so heat stress affecting seed affect not only the formation and the quality of the grain, but this will induce sterility, produce damage in the rice yield, and affect fundamental processes like germination, fertilization, etc., etc. So one of the things we did was start to analyze to this literature, and we found, for instance, that there were proteomic analyses highlighting that, for instance, many late embryogenesis abundant proteins were upregulated in terms of protein accumulation in these varieties of rice that were exposed to heat stress. Okay, so one of the data we did at the beginning of the project was to study in detail the function of these genes, since, for instance, it was reported that these LEAs, or late embryogenesis abundant proteins, are important proteins with multiple functions controlling osmotic stress. For instance, they can act as molecular chaperones that are able to stabilize proteins and also can prevent the naturalization or aggregation of proteins that are exposed to osmotic stress. So among the upregulated proteins that were found in those papers, we perform a in silico analysis of expression, taking advantage of transcription data already available. And for instance, we identified 3 different late embryogenesis abundant proteins that were specifically expressed in the grain quality or in the grain milling formation. So those specifically expressed in embryo development, in endosperm development, and that also were upregulated in the proteomic studies on heat stress. And we clusterized them in different groups, for instance, identifying that they belong to the same group, the group 4. So they were very good candidates to perform functional studies, okay? So I will not explain in detail the different pipelines, just consider that the entire process is quite long because, for instance, the generation of constructs, the transformation, regeneration, and the validation by sequencing, it's a process that sometimes requires several years, since in many cases we find always the same type of edition or non-edition, depending on the importance of the gene, of the target gene. And we follow all this procedure, okay? In concrete, one of the key steps, which is the transformation and selection, can take from 4 to 8 months. And for instance, it's very important to regenerate proper calli that later will form the future plantains. So just providing some guidelines of the results until today in the C4C project, we were able to generate single mutants for the 3 layers we were testing, although they are in different processes of validation actually, okay? And with the first already gene edited and confirmed, the layer 14, we were able to use this as a background of new transformation protocols for the other single layers in order to be able to generate double and triple mutants in a faster way. So this is one of the cases or of one of the lines that we have generated. For instance, a gene-edited line for layer 14 that contains a 26 per base deletion in the second exon of the gene. And for instance, one of the validations we have done in relation to what was discussed before is the existence of targets. So for instance, to validate that this line has none of targets, we selected the 3 most potentially of targets respect to this LIA, and we in fact confirmed that there was no presence of target mutations. One of the interesting characters was found in these lines. For instance, this line presents severe defects in growth, pale leaves, and also a reduction on the leaf length. And we found also that this impact of the mutation affects also productivity. For instance, has lower number of panicles, and also the seeds that are produced are shorter respect to the wild type situation, confirming that this function is important for the overall development of the plant. Okay. In concrete, we focus our attention on the seeds, for instance, that were smaller, and we perform some characterization, for instance, on the dynamics of germination, where we found that in the mutant there was an important delay in germination, as you can see, versus the wild type, that germination occurs many hours later respect to the typical wild type germinating along or starting germinating after 2 days in a control medium. And also the dynamics of germination, apart of delays, provoked that there was an asynchronous root and shoot emergence with aberrant dynamics in secondary root emergence. For instance, the mutant had a synchronous emergence of the root and the shoot emergence, which is not the typical case in a wild type situation. And also it hasn't the presence of secondary root. So then we also are starting to study the impact in drought stress. For instance, with this mutant, we know that it is hypersensible to drought stress tests. And we have also done some expression analysis to configure the potential role. For instance, this layer 14 is highly upregulated short after the formation of the grain, so at 4 days after pollination, indicating that this layer potentially has roles at the beginning of the formation of the seed. And with this result that we have presented today and much other that we are generating with the progression of these lines, we have different key messages on the C4C results. So we can confirm that layer proteins are importantly transcriptionally regulated under drought stress. We are generating single and double mutants. They are potentially involved in seed development. And this is affecting also germination and the overall development of the plant. We need further investigation because there are many aspects still up to date and concerning the role of the layers in different developmental processes and in relation to abiotic stress, and additional tasks apart of that well presented during the talks. So I want to just to have 1 minute introduction of one extra project that is actually have been started in my laboratory, which is a part of the C4C project, but is quite related to this thematic, which is the future grain. This research aims to explore rice diversity in agricultural systems and controlling natural nutritional quality towards a healthier food with a lower environmental impact. So for these studies, which are focused on biodiversity of rice, we are collaborating with the Piemonte Orientale University, one of the universities in the north that are more focused on rice, and also with the agrarian department of my university. And we are doing a screening on different varieties of rice. We start from 340 ancient and modern varieties of rice that have been cultivated in the Mediterranean basin, so from Spain, Greece, Turkey, and France and Portugal, and we have screened them for stress tolerance to salinity. We have created a subcluster of lines, 40 different lines, that are actually under investigation for different productivity characterization, the impact of drought stress in these lines, the interaction with plant growth promoting bacteria, and other aspects that will potentially give us more clues about new gene candidates that are controlling drought stress tolerance. So with this last slide, I would thank you, all of you, for your attention, and also especially for the BATEM colleagues for hosting us, for the nice and wonderful views of Antalya, and for the very wonderful food. Thank you very much.
Any question? So just the edited plant is rice is Nippon or
yes, it's Nippon Bare is the line we use as for this type of functional studies, because it's quite short in terms of life cycle, quite transformable because we generate quite easily calli from the seeds. And we have a long history of control of growth conditions, which is very critical, because we have Il Brusone, which is a very important pest in greenhouse for rice. Okay.
Okay.
Thank you very much.
Yeah. Yes. So now there is Kamal Abdekani that will do presentation about study of the effect of water shortage on the physiological and agronomic parameter of various genotype of eggplants selected for drought tolerance grown under the Mediterranean climate condition of the northeast of Morocco.
Thank you. Thank you very much, Sipe. Thanks for the Turkish partner to be here with you. Thanks for C4C. That's why we are here, and also to continue our collaboration in the future. So today I'm going to present actually two subjects. Probably I should change a little bit the title, but I'm going to present the genotype of eggplants in the C4C project, and also I'm going to talk about another project which is zero tillage of the cereals experiments done in Morocco. Okay, so yes, can I change? How do you change? Okay. Okay. So yeah, for the introduction, I will be a little bit fast in my presentation. So we had during the last 6 years a severe drought in Morocco, I can say like we had like 60% of reduction in the rain, depends of the region. So it's like kind of a climatic drama for us. So even we have like ambitious plan, which is the green Morocco plan. So we are really facing this problem of water. So now research and development is focused more on the water management. And when we talk about water management, we talk about integrate solutions, including breeding. And I think breeding is an important, component of this water management strategy. So yes, we need this kind of work, not only for science, but also for deliverable for the growers. Okay, which kind of seeds that we can plant and when, and especially the season, they are very beginning very short. So it's important to choose which kind of seeds. So I am going to present for the first part. Sorry, here it's not direct seeding, but it's the C4C project. So the main objective is, I will go quick. We had we test genotype from BATEM. I think probably it can be the first time in this area of the Mediterranean northeast of Morocco, and we test also some genotype of CREA from Italy. Other genotypes, commercial genotype also that we test in commercial, used by the growers. We ran 3 years experiments. Really, I can say a very hard field work. We collect a lot of data. So if you can see here, like the first, year, 2021, 2022, where we test here on the right, we have 100% of water, 50% of water, and this one is 100% of water. And when we talk about 100% of water, that's mean the irrigation made by the grower. It's not by the evapotranspiration. And the second year, as you can see here, the first year we had 15 genotype, including CREA, BATEM, and commercial, varieties. In the second year, you can see the design is a little bit different. It's more genotype that we test. Some genotype were repeated. And also we include another solution, which is using biofertilizer. So because the biofertilizer, it's according to literature, the biofertilizer can help plants to tolerate against stress. The question here it's which kind of biofertilizer we can use. In this case, we use a fertilizer based on Pseudomonas, which is it's in the market, right? So because we want to make sure that this biofertilizer work. Here I'm not showing all, it's the concept. I am presenting the concept, okay? So each plot here in 2022, 23, it's 4 plants, okay? So we have enough repetition, okay? And the third year we use in a small, this is the same site, and this one is, we use the evapotranspiration as a control for irrigation. We take many parameters, water consumption, plant growth and biomass, relative water content, stress index. This is, I'm going to explain a little bit very fast the concept of this stress index and the plant yield, fruit quality. Now when Dr. Rotino, he was talking about which index that we should take in consideration. So this is the question, because just in this stress index, we can measure like 60 parameters. Probably, I don't know if you are more familiar with this tool, but we dark adapted our leaf for like 30 minutes, okay? And we give a flashlight, like 3000 micromole in order to saturate our photosystem 1 and 2. And we imagine we do this measurement, those clips. I have 120, so I split them on all those plants at the same time from 10 o'clock in the morning until 4 o'clock in the afternoon, in order to know different parameters that I can show here. Okay, before the concept is this spider model. So each parameter is an index. we can, if you back to the literature, each parameter can give an instantaneous measure of stress, and others they can give a long-term stress. Okay, you know, like I think 3 years it's not enough to analyze all those data of fluorescence, because we take this measurement each month and each 3 days, typically days, hot days, from 10 o'clock until 4 o'clock in the afternoon. Okay. So the concept is here, sorry, it's in French, but anyway, I think it's clear. Here you have the photosystem 2, where you have a normal plants. So the reaction center, they are open, and when they are stressed, they are closed. So imagine if we give a flashlight, so you have one part is converted for, you know, photosynthesis for the Calvin cycle, and one part is going like as a fluorescence, and the other part is going like as a heat. So with this tool that we have, you know, we can estimate what's going on in the light reaction. So those one, they are really a good indexes, and we did it for all the genotype for the 3 years. Okay, so we have a lot of data to analyze, and probably we can get something interesting from this. Okay. Here I will just go quick for some data. I'm not going in details, but as I said, the yield is, I think, is the most important parameter to take in consideration. And we have, yeah, as I said this morning with the group, we have to be careful when we conclude about the results, because you have so many parameters. So we have to conclude about what we have in this field. Probably if we repeat it in another places, probably will not get the same results. But anyway, so we have different response of the yield. One of the preliminary results, we can say that with the biofertilizer, we can enhance the plant tolerance for the drought. Another most important thing is the characterization of the genotype, how they behave under those climates. So you have like 10 accession of CREA with 4 of BATEM, and also with the native, some native seeds from this area. So the response is not the same, okay? And also when we say the water shortage and water scarcity, they are different definitions, okay? And also the water scarcity depends when you cut your water. In my case, I cut water for all the phenological stages. So when we say this water shortage, it's another issue that we can take in consideration, because you can stress your like plants during the flowering, that as you mentioned, but in order to enhance the productivity or to get more big fruits, you know, or more yield. Okay. Anyway, I will go through this very quick, but those experiments, they are done for 3 years, and we are in process of analyzing them, and also to publish those data for the next years. Now, I am going to present another, I would like to thank my PhD student, Dina Maeshi. She's not with us, she's with us online right now, but she did all this work in the field. So she collaborated with us. The second work is about cereals in the same area of Morocco. I will try to be very quick in this. We are working on the zero tillage. You know, zero tillage, that means we seed without working our soil, okay? And we have a national program until 2030 to plant 1 million of hectare of cereals, okay, with using this you know, technique. And I think we will have a good research in the future to develop this concept. According to literature, we can have like 10% more production, less water, less gas emission, and again in the decarbonation, like in the carbon. Okay. This is some picture. Inyasur, he was with us in this area, I think in 2 years ago, because we are searching about to develop or to test new varieties or genotype of cereals, wheat, and durum also, to see the tolerance for under those conditions. Okay. Here we had the control and the treatment, direct seeding and the conventional seeding. We use 500 hectares as a platform because we are collaborating with two teams in CSIC in Madrid about artificial intelligence or the teledetection, because we take GPS codes, soil analysis, and the growth data, and we want to develop kind of forecasting the production for the next years, and to use this tool with probably with also using different genotype in order to see what's going on, and also with the climate condition. So imagine we take all these parameters. This is my student, master student. He finished on this area, as I said, in the northeast of Morocco. So we take all those parameters, crop yield, plant biomass, and etc. Okay. Here's some, we did the destructive measurements of the plants in order to see what's happened in zero tillage and, the normal seeding. As I want to mention that some preliminary results show that in the direct seeding we have better yield in SD, which is direct seeding, and conventional seeding. And also we measure that, this is in French, semi-direct, which is the direct seeding. We have a bigger, I can say, seed in this, in comparison to the conventional seeding. As I said, we take many parts, and we are trying, and with GPS, and we are trying with the remote sensing to predict, the production for the next years, and also to develop a modeling, evapotranspiration model. We follow also the NDVI, which is measured by the Sentinel-2 for those crops. You can see here the NDVI of the conventional seeding. So it's an index of the vegetation. You see, so you have more vegetation in the direct seeding comparison to the normal one. This is an interesting map that we tried to do an experiment, but we had some technical problems. But the concept, what I try to do here is with in collaboration with l'INRA, which is BATEM, or CREA's Moroccan, National Institute of Research in Agriculture. So they give us some seeds. In here it's like without irrigation, and this one is with irrigation. Okay, and we have 8 varieties. Okay, so we want to compare between irrigation factor, genetic factor, and also with direct seeding and without direct seeding. And we want to use a drone with multispectral camera in order to know the coefficient, water status, vegetation, and other indexes, and how the plant behavior we use in this tool. So yeah, this experiments actually it failed because, as I said, for technical problem, because we didn't have water. Imagine we have like 100 millimeters of water in this area, so no water, and even to irrigate we did not have enough water. But we will repeat it again, and why not with the other seeds and other genotype in the future. So yeah, we use those platforms with, of course, with students, with master student in plant science, and in large scale field. So yeah, so it's kind not only for research and research development, it's good for the stakeholders, because we share with them our results, and also it's kind of education for the next generation of scientists. Thank you.
Yeah, there is one question.
Very nice talk. I was wondering about this direct seeding strategy that you are testing in cereals, because as far as I remember, this requires genotypes that are good for direct seeding, and also change agricultural practices on soil. So do you think if your research progress, you can convince farmers to adapt these strategies?
Yeah.
Which are the challenges for that?
Yeah, good question. I think because before, like 20 years ago, when we had much rain, because we are in the semi-arid area, 500 millimeters, growers, they start seeding like in November and harvest in, in June. But now with this climate change, the seasons change. Sometimes you have rain, sometimes it's cut, but I think seasons, they get shorter. So it's good to adapt or to develop kind of new varieties with a short period where you have like this water availability, and also some seeds adapted for this new tool. As I said, Morocco now put funding for in order to achieve 1 million of hectares in 2030 with using this tool, because it's also good practice for the carbon sequestration, because we will have like more credit on this. So I think the focus for the next proposals is to search the adapting varieties for this climate condition and also for the soil characteristic.
Because it's less damage less the soil,
yes,
the physical chemical properties of soil. So
yes,
it's a very good practice.
Because with the intensive agriculture, we destroy our soil with using these machines, and also using the artificial remote sensing is good because we can know where and when we can take the decision to seed and to plant. So if we combine the the remote sensing with the breeding, it will be a good tool and a good deliverable for the growers.
Okay, thanks.
Yeah.
Yeah, thank you for the presentation. Really fast and short question. For the second year, what is the plan for sowing seeds, like overseeding or minimum tillage before seeding?
The question, sorry?
Like you mentioned, you are focusing on minimum tilling.
Yes.
Before seed sowing. For the second year, what will you gonna do? Like overseeding or minimum tillage and after seeding new seeds?
No, because if I understand well your question, we usually growers, they use the direct, I mean, the conventional seeding. They work the soil, or just they seed like this in those area with the hand.
Yeah, then you are not planning to remove previous year residuals.
Yes.
Okay, then.
Yeah.
Overseeding.
Yeah.
Okay, thanks.
Okay. Thank you.
Okay.
Nesline?
Yes, I'm here.
So the next speaker will be Sally Nesline from Algeria, and she will talk about a response in germination and seedling growth in some Algerian durum wheat under salt and drought stresses. Here she is.
Yes. I can share my screen, please.
Söyleyebilir misin? Açıklayalım. Bağlanamadı herhalde de. Aynen. Önce PowerPoint'i haline getirecek. Tamam. Ondan sonra
You cannot share the screen?
Yeah, I didn't.
First of all,
Possible, yeah.
Open your presentation full ekran and then share.
Yeah. Yeah, it's open, but saved.
Please choose window appearance. Run screen.
Yeah, okay. Wait a minute. I have some problem. Is I don't know.
Nesrin, no problem. Go ahead. We are listening you. We are listening you. Okay.
I'm back. Sorry.
So Nesrin, I think maybe it's better to go ahead just talking. You can make a fast summary of your work just talking because the connection go down.
Okay. You are see my screen? Are you see my screen? Please.
Yeah.
Yeah. Are you see my screen? Okay. Okay, thank you everyone, and Assalamu alaikum. Thank you for the BATEM for give me this opportunity of my PhD student Najat. It's titled Response in Germination and Seedling Growth in the Some Algerian Durum Wheat Under Salt and Drought Stress. So I start with this phrase, thinking globally and acting locally. This it was born early in 70 by the environmental person to encourage the individual to improve their impact on environment and through the local action. This is for the people, for the plants, for the world problems such as climate change and environmental degradation, like the desertification, have raised alarm in the world. Weed do their effect in the biodiversity and agriculture. In the Saharan region, in the Saharan region, war conditions are becoming progressively existing due the global warming remain that the valued area and the soil quality or severe climate condition. Cereal cultivation and specifically wheat is the food, the important food or source of foods in the people. In the world wheat and played a critical role in agriculture, system agriculture, in the economical important and in the nutrition of human. Algeria is cultivate a large scale of cereal. The wheat is important crop in Algeria, was cultivated by upper to 3 million hectares of area of Algeria is cereal. But this area is affected by different abiotic and abiotic problem or stress like salinity and drought. This is affected in the loss of yield or yield loss in this region. Salinity and water stress has affected the different stage of plant development, like germination or seedling or flowering stage. For this reason, the important to develop different process to selected some variety tolerant on the salt or water or heat stress. The aim of this work is to identify or key morphophysiological parameters that can be used as simple and fast selection critical for abiotic or abiotic tolerant durum wheat genotype. For the material of this experiment, 60 local genotype was selected for first experiment of germination, and this 60 genotypes was exposed to different stress like drought stress, heat stress, and saline stress. And then we measured the different parameters like germination, percentage, root length, leaves, and leaf area. Sorry. For the second experiment, is the experiment of field. We tested these fourth genotypes in the different area. In the different area or Saharan region. It was characterized by saline irrigation and by saline soil and water and saline water irrigation. And the second area, it's characterized by acceptable area for the salinity of soil and water. In this experiment or field experiment, we tested the combined stress, like saline stress with water stress or drought stress, saline stress with heat stress, saline stress with drought stress. Concerning the water stress, we applied the water stress in the beginning of the ending of the beginning of flowering stage. The different parameters we measured, like the 1,000 grain weight, yield, number grain, chlorophyll content, and morphometry measure. And we measured some ecological index. For the result for the first experiment, for the I don't know if you hear me. Are you hear me?
Yes, yes.
Okay. Now for the results of this experiment or the germination experiment, we observed that the drought or the drought has a significant difference effect in the germination speed among the tested wheat genotype. Like the Bukhalouf, Fritisi, and Fartas, and Hidba, Vitron wheat, Vitron Waha, and Chance is more tolerant to drought to drought stress. But the other genotype was present a sensitive or drought stress. In the second experiment, we selected only 8 genotype to test the stress or saline stress. We observed, or in this study, we compare the response of the different durum-wheat genotype to salinity stress, and we measure the several morphological parameters. For the root number, or length, leaf length, and shoot length, and leaf area was observed. All results indicate that the different genotypes variety has a very level tolerance to salt stress, as the evidence by the different response in terms of root or shoot or leaf growth. For the heat or temperature stress, we tested only 4 genotype in the high temperature is 45 degrees. We observed that is not all results is not a significant effect on germination or germination percentage in the heat temperature. For this, we can said the temperature is not significantly impact in the percentage of germination of tested genotype. In the further result of field or result of combined stress, we observed that the combined stress is like saline and drought generally is lead the most sensitive reduce in the plant high compared to signal stress. Bukhaluf, Ifritisi, and Hidba were practically affected. On the other hand, all the parameters was affected, but by the different valued to compare with the signal stress. This result in this we summarize all the results in this figure. Under the combined stress like drought stress and drought temperature, drought and salinity, and drought and temperature, the fourth variety followed a multiple tolerance strategy with some similarity. They all reduce their life cycle, seed size, yield, and number of seed per speak, but they showed a difference in method of absorption of accumulation of sodium. We synthesize this by these results by B-plot the row. We observed that the strategy one with the the different genotype, like the HEDBA in drought temperature and FRITC on drought temperature was same strategy, but is a different totally to بخلوف in drought and بخلوف in temperature. Also, the different or the sorry. Also, the index of the index of ecological index the present the strategy of the different different genotypes or studies genotypes. We observed fourth strategy like FRITC and drought and FRITC and salinity and FRITC and بخلوف in salinity was a same or opposite by بخلوف and in the drought and temperature stress. In the conclusion, or this study shows the four local population studied were able to tolerate abiotic stress with the different strategy, but this lead the similar results. Morphological parameters and yield parameters are not significant to detect the best variety that can tolerate specific condition. The index or ecological index have population with a high salinity and drought, with qualify them the suitable for selection and high yielding genotype under both stress. This study population have shown considerable variability in yield and stress tolerance, which could be exploit the crop improvement and a right choice, the variety or genotype for the different field condition. And finally, we will come in Algeria with a diversity of local there is sea and sun and thank you for your attention.
So thank you, Nesrin. There is some question. Yeah, there is Nacho.
Hello, Salhi. Thank you very much.
Yeah.
Very interesting talk. I want to ask you about the genetic diversity on these four varieties you are testing. So if you know a little bit about their, let's say, evolution in origin or the genetic origin, or if you plan to do some characterization of the genetic profiles of these varieties.
For this fourth population, local population, is it's not in this moment. It's not. I'm not confirmed. It's the pure line or the only the hybrid population, but I in the local agriculture since 1000. This local is this population of this genotype is adapted locally in the condition of the Saharan condition. But for the study, genetic study is not yet, because in Algeria there isn't and some capacity building to make this kind of this study, and I hope to in the future to make any funding to approfund in this study, genetics study of this local population or local genotypes.
Thank you.
Yeah, come.
Yeah, thank you. And just to, how do you think to exploit this adapted genotypes, local population? Do you think to enlarge the cultivation area in Algeria?
For this four, the sixth the 16 genotypes is cultivated in Algeria, but for the only the fourth genotypes or local genotypes is cultivated only in the Sahel region, or specifically Wad Rir region. It's Wad Rir is characterized by high salinity of soil and high salinity of water. It's salinity of soil, it's of water, it's equal to 9 grams per liter. It's a very high saline water.
Okay, thank you very much.
Yeah, welcome.
And yeah, I think we can skip the conclusion or not. Yes. So I thank all the speakers, because since people is quite tired for this long day of work. And I thank again everybody for participation. Hoping to meet again in presence, hopefully. Thank you again.
Thank you all. Thank you all, and
bye.
Thank you very much. Bye-bye.
please for tomorrow